Okay, so we're going to talk about enzymes for today's notes. Enzymes are basically employed proteins. That's always the way I think of it. So there is a particular flow of energy throughout life. Life is built upon chemical reactions. So the giraffe gets its energy from the plants. The plants got their energy from the sun. So from the sun to the plants to the giraffe. And then the lion gets its energy from the giraffe, who ate the plants, who got energy from the sun, aka ecology. But all this happens due to chemical reactions. And in these chemical reactions, nothing works without enzymes. So how important are these enzymes? Well, all chemical reactions require enzymes. There are synthesis enzymes, which are responsible for building. To synthesize means to make. There's also digestive enzymes that breaks things down. And there are enzymes that speed up reactions without being used. And those are called catalysts. For example, the liver lab we did, catalase enzyme breaks down hydrogen peroxide into water and oxygen. But that enzyme is reusable. You can use it multiple times. So an example would be dehydration synthesis. So the enzyme will go in between these two molecules, take water out to build a bond. An opposite example would be hydrolysis. Hydro meaning water, lysis meaning to split or cut. So hydrolysis would take the enzyme and the molecules that are joined and do the opposite. It would break it apart using water. So we'll talk more about this later, but dehydration synthesis takes two things and makes one by removing water, and then hydrolysis takes two things and splits them apart by adding water. I always think dehydration like making beef jerky. You make beef jerky by dehydrating the beef by taking water out. So in order to do these reactions, we have this thing called activation energy. Activation energy is the energy needed to start a chemical reaction. So if I have a bonfire, the fire doesn't start till I input some energy. And it could be a small fire starter, a small bit of flame added to this massive pile of wood, but nothing's going to ignite till that starts eating and getting bigger and bigger until the whole bonfire is lit. So, reducing activation energy. Catalysts make it easier for these equations to occur. So it reduces the amount of energy to start a reaction. So you have a reactant, which is your starting thing, and then your product is what you produce. Easy way to remember this is you don't go on the show Shark Tank pitching your reactant. You pitch your product. You pitch what you make. So reactant is the start, product is the end. But in order to get there, there's energy needed for it to occur. If you add in an enzyme, which is a catalyst, it lowers the amount of energy you need, making it easier to do so. You could walk to Harrisburg. Might take you a while, but there's a bridge. There are paths. You can walk there. I do not recommend it. Or you can get in a car. The car is reusable, though. It doesn't get to Harrisburg, drop you off, and then the car just, bursts into flames. The car is reusable. The car is like an enzyme. It speeds up the reaction. It speeds up the time between A and B, but it's not used during that reaction. So enzymes are proteins. They are a biological catalyst, meaning they catalyze living reactions. The thing is, though, each enzyme works for one reaction. It needs to be the right shape for the job. Think of it as like a lock and a key. Your key only really works for one lock. Even though they're all keys and all locks, they're each shaped differently. The enzyme they are named for, the reaction they helped. So if you have the enzyme sucrase, it breaks down sucrose, which is a sugar. If you have protease, it breaks down a protein. If you have a lipase, it breaks down a lipid. If you have DNA polymerase, it builds DNA. Notice they all end in A's, just like almost every sugar ends in OSE and ose, right? Glucose, sucrose, fructose. A's is an enzyme. Things that end in A's and that sound are often enzymes. So some facts about these. Enzymes are not changed by the reaction that they catalyze. They are only used temporarily. They can be reused again for the same reaction with other molecules, and very little enzyme is needed for those reactions. You had your piece of liver in the liver lab, and you could keep adding hydrogen peroxide to it, and it kept going. You don't need much. Any little bit will do. So here's our enzyme over here. This big purple chunk is our enzyme. The active site is the site at which the activity occurs. It's where the thing is either broken down or built. The substrate is another word for reactant. So if you see substrate or reactant, they mean the same thing. And product means what's being produced. So in this case, your enzyme stays the same the whole time, but as the substrate or reactant comes in, it fits into the spot, the enzyme breaks that bond in between them and puts out the product. But that enzyme is not changed. It can be reused and reused again. So it's a shape that matters. We call it a lock and key model. So the key comes into the lock, turns, releases the product, keeps going. So this is sucrose going into the enzyme. The enzyme adds water to break it down. So it's hydrolysis. Water is being used to cut. It's cut into two parts. Sucrose is made of glucose and fructose. And then it's ready to go again. That active site can accept something else as soon as it's gone. So some vocabulary that is particular to this, and some of this I've already said, the enzyme is that helper protein molecule. The substrate is the molecule the enzyme's working on, also called a reactant. The product is what the enzyme is making. It's the end result. And the active site is the site at which all of this activity occurs. It's where the enzyme breaks down whatever it is breaking down. It could be sugar, doesn't matter. So some things affect enzyme action. It could be temperature. Why? Because, well, if you cook or heat up an enzyme, it unfolds and changes shape. And since it's not the right shape anymore, it doesn't work. If you torch a lock and melt it, of course the key's not going to fit. pH, acid and bases, same thing. If you drop a lock in acid, the key's probably not going to work anymore. Everything has to be the right shape, and both pH and temperature can change the shape of the protein, which would then make it ineffective. So temperature in particular, there's an optimum temperature, optimum meaning preferred. So for human enzymes, it's our average body temperature. If your body gets too hot, your proteins can start melting. So an extremely high fever is dangerous because the proteins of your brain, your muscles, can literally start breaking down. So that's why anything over like 106, you go to the hospital for. Raised temperature, boiling, you denature or change that protein, it unfolds, it loses its shape. Lower the temperature, they move too slowly, and then they're not gone, they're just not efficient. So there's an optimum temperature at which enzymes prefer to react. Can they react higher and lower? Yes, but they work best at a certain temperature. Changes in pH change protein shape. So most human enzymes are a pH of about 6 to 8, but it also depends on where in the body. So pepsin, which is an enzyme in your stomach, has about a pH of 3. It's very, very acidic. Whereas your small intestines have a pH of 8. They're very basic. Your stomach has a lot of acid in it, but when you go to the restroom, you're not, your waste isn't acid because the acid is neutralized as it goes through your intestines. Your intestines are very basic to help neutralize that acid. So what that could look like is the bell curves are different. So for your stomach acid, the enzymes work at a lower rate. They have a lower preferred or optimum pH. For the normal, they're looking for about 7, but your intestines might have a higher pH that they prefer. But what matters for enzymes is shape. Form determines function. The shape of the enzyme tells you what it does, And the shape of the enzyme is what allows it to do its job. If you change the shape, it does not work. So kind of sideswiping away from enzymes, just to go back to water. Water everywhere, right? So water is important. Water is a molecule that is used in these reactions. It can be made in these reactions, and it has its own reactions. The drops of the penny lab shows that water sticks to itself with cohesion. Oxygen has a negative charge, while hydrogens have a positive charge, which allows them to make weak associations, negative to positive to negative to positive, and they build that kind of relationship, which is strong-ish, but can be broken. Now, water has 6 properties that make it very, very useful and essential to life. 1, they're polar. That's what I was saying. They have a partially negative, partially positive, and they just keep lining up with each other. Cohesion and adhesion. Cohesion is water sticking to water. Think cooperation. Adhesion, think about tape. Adhesive. Tape is because it's sticky to other things. Adhesion is water's ability to stick to other things, like water droplets on a leaf. They stick to that leaf. Water has different densities, ice versus liquid. So ice floats, right? Have you ever seen a pond and wondered why the fish don't die every year when the pond freezes? Well, it's because ice floats. And it freezes just the top of that pond, which actually insulates and keeps the bottom of the pond warmer. It's kind of like an igloo, of how an igloo keeps people warm. Keeps them warm because it insulates. Ability to hold heat. Water can store heat. So if you have a pool and the sun's beating down on it all day, the pool will warm up because it can absorb that sunlight energy. It's a universal solvent. So solvent just means it dissolves anything. Just about everything will dissolve in water if you give it enough time. If you think about the Grand Canyon, the paths and the channels in the rock were carved by water. So water can do stuff. And then capillary action. That is how like water moves up a tree. Is water can get pulled from area of high to low concentration. If you thought about if you have a paper towel and you put water on the corner of it, the water will absorb through the paper towel. That's capillary action. So all these notes are for you. Make sure you have them down. When you're done, make sure you click submit. Thank you for getting this done, and always email me if you have any questions.